Well, thank you very much. And it's a great pleasure to be here today and to tell you a little bit about some of the things that we've been doing in my laboratory. I should mention it is biological sciences that I'm in. I don't know that I could get away with studying all these creatures in the School of Medicine, but they do inform us about a lot of different facets of general features about the way animal brains and behaviors sort of interact and evolve together. And so what I'm going to do today is try to showcase some of these different species and tell you about some of the amazing things that I think are their abilities to explore their environments. A lot of what I want to talk to you about is probably going to be in a predator-prey context. So, you know, the challenge of how do some of these animals either escape from predators or how do they prey on other animals. And there's a lot of very interesting specializations. And throughout, where possible, I'll try to point out some of the things that we've learned about nervous systems from these species, because they also inform us about how brains have evolved. And sometimes specialist species can really give us clues into important ways about different nervous systems function. So if you were really interested in the sense of touch, you might look at a touch specialist. For example, if you were interested in the sense of hearing, you might look at an auditory specialist and those kinds of themes. So I'll mention a few of these as I go along. I'm not going to talk about all of these animals. This is a partial cast of characters of the creatures that we study in my laboratory, but I'll talk about star-nosed moles and water shrews and electric eels and tentacled snakes. They're all very diverse and very interesting. And you know, there's different reasons why you might study specialized species. And you know, the theme that you might have, of course, and if you're writing a grant proposal, is learning about general principles, about animal behavior, about nervous system function, about brain organization. We can also learn generally about nature and teach about nature. So the animals, I think, are intrinsically interesting in their own right, and are fascinating in that respect. And then sort of the other secret side of it is it's lots of fun to do this kind of research. So, right, so can't help but mention that. So the area that I'm in is often called neuroethology, and that's a combination of neurosciences and animal behavior, trying to put those two things together, which is not always the easiest thing to do, and especially when you're interested in unusual animals that may be either hard to find, hard to keep, hard to study. And so that's some of the species that I'm going to talk about. And I'm going to start off talking to you about this guy, which is the North American water shrew, a very interesting animal. It looks like a mouse, right? But it's very distantly related to mice. It's not related to mice and rats. It's an insectivore, so it's a shrew. And they have historically been considered to be sort of very simple, primitive animals that can't really do very much with tiny little brains. In fact, you know, if you compare a shrew brain to a human brain, here's the little shrew brain on a penny, right? And here, this is a more familiar brain that hopefully we all have, right? And so clearly you might look at this and say, well, this animal probably can't do much, and you know, we've got skyscrapers and cars and drones and all kinds, and cell phones, and so we're more impressive. So I want to tell you some things that this animal can do. And I'm going to start with telling you that I think of it as sort of the great white shark of its habitat, because it's a semi-aquatic mammal. You might guess from the name water shrew that it swims. And it feeds on lots of things. In fact, it's very inquisitive. It'll explore new environments, and it will try to eat anything that it can overpower. So here's one of its prey items. And you know, these crayfish can be pretty intimidating for a small mammal. I should mention that these shrews are quite small. They're smaller than mice. They're about 15 grams, so they're really tiny. And they swim underwater, and they will definitely go for this kind of prey. And this looks really intimidating in terms of trying to deal with something like that that's armored with these sort of 6 guns drawn, right, ready to defend itself. And I'll tell you the way this ends every time is not good for the crayfish. And it's basically not fast enough. And that's one of the interesting themes about the evolution of different nervous systems and the behavior of different species. This is a warm-blooded mammal with a body temperature that is greater than the environment, typically. They live in North America, though they do come down into Tennessee and the Smoky Mountains. So they don't hibernate, they're out in the cold. In times when these guys are much slower in general, even during the summer, this animal is going to have the body temperature of the water. And there's another innovation. I'm not sure if others have spoken about this, but mammals have myelinated nerve fibers. There's a fatty wrapping of cells around our nerve fibers, and this speeds the conduction of action potentials, the fundamental signals in our brains, very greatly compared to many invertebrates, which can only speed things by getting their nerve fibers to be huge. And in fact, crayfish do have giant nerve fibers that they use to escape from predators. They send signals to their muscles to contract for escape responses. But despite having these large nerve fibers, the mammal myelinated fiber, even though it's much, much smaller, is much faster. It's about 3 times faster than a crayfish nerve fiber. So that's one of the reasons that the crayfish just doesn't have a chance against something like this small mammal, at least on land. Now, they have a much better chance underwater, and it's amazing to look at what these shrews can do underwater. They'll, as I said, chase different prey. They can't dive for long periods, but they do dive for most of their food for short periods because of their high metabolism. It's only about 20 seconds. But in that 20 seconds, they can do a lot. So I'll just show some imagery because I think it's fascinating to think about this animal, which has small eyesight, swimming around at night. It's nocturnal. So imagine trying to find your way around forests, streams, the edges of creeks, and then dive in to get prey at night. And they can do this very well. And they can do this also to catch fish, which is remarkable because fish are really fast. So I'm going to expand on that later in the talk and actually show you some details of the fish escape responses. But let me start by showing you what a water shrew can do. Whoops, hopefully this will. Do we have the movie up? Doesn't look like this movie's going to play. Oh, good. Okay, this is slow motion, and I should say it's infrared lighting, meaning that the shrew cannot be using eyesight. They can't see infrared light. Now, see this in real time. That was slow motion. Really fast. You'll see that again. Now, you see another example. And one of the things I want to emphasize is this animal's keying in on the movement of the prey, using its whiskers. So that's a clue to what it does to find prey. Now, watch this in real time. Now, that is one of the things that's unappreciated about small brains. So small brains have connections that are relatively short. All the cortical areas are very close together, which means processing is very quick. And if you think about computers, what are the great advances? You know, when you want to get a new computer, what do they always say? The processing speed. Oh, it's so much better than the last computer. Well, these guys have very fast processing speeds, and this gives them a leg up on some of their compared to other mammals, and that's a theme I'll also mention. Very high-speed behaviors is something that a lot of these animals have. I do want to mention a little bit about their brain organization. So they have neocortex, a fundamental characteristic of mammal brains, and neocortex contains maps of sensory surfaces typically. And this animal's neocortex is dominated by what's called somatosensory cortex or touch cortex because the whiskers are just so important to that animal. And in some species, you can actually see these brain maps that are normally sort of drawn in sort of this diagram. You sort of, well, you have to believe, okay, I'm saying that there's touch information processed in this part of the brain. But if you look in the lower part of this animal's brain, on either side of the brain, you'll see these sets of circles. You see this almost looks like a compound eye of an insect, these little dots here. Each one of those dots is where one whisker projects from the shrew's face. So you can see where one individual whisker information is processed. And this is really extraordinary to be able to see the brain maps in different animals. And I'll expand on that as well. And it's a very useful thing for studies, and it's also very interesting just to see. So this animal also has another amazing trick. I want you to watch this because this was thought to be impossible for animals. That was underwater sniffing. So it was thought that no mammal could smell while it was underwater, because how could you possibly do that? The way this shrew does that is to exhale air bubbles onto things it's exploring and then re-inhale the same air bubbles that it exhaled to pick up odorants. And so this is an amazing workaround for olfaction for a swimming mammal. Everything you just saw there was extremely slow motion, right? So they do this very quickly. Small mammals in general sniff very quickly. So the only way to discover this is to put them under the high-speed camera at about 1,000 frames per second and then film them underwater. So this is something we've only recently discovered. But I just wanted to emphasize, some of the amazing abilities of these animals, because when you think about, could this animal just be primitive and simple, it can do some things that we could hardly have imagined until we looked closely. And using this underwater sniffing ability, it can follow a scent trail underwater. So now you'll see this is also slowed down, but it's a little bit faster. And this is a color shot of it sort of zoomed out. And what I want to tell you is there's a scent trail going in this direction, but not in this direction. And you'll see that the animal is doing this underwater sniffing behavior to follow down the scent trail. And basically, it's very comfortable underwater. It pops to the surface when it stops swimming because its water-resistant fur traps air. And there's a food reward there that it just got. So it sniffed down the scent trail and got its food reward. So pretty remarkable creature. Now, in terms of underwater sniffing, this is not the only animal that can do this. Now, here's a really very unusual creature called a star-nosed mole. It looks like it's from Mars, but it's actually also found in Tennessee, in the Smoky Mountains. But most of its range is in northern North America, all the way up into Canada. And it can also do this trick. It's also semi-aquatic, like the water shrew. It lives in the same tunnels. Goes underwater occasionally to search for food. And here you see those air bubbles being extruded. And of course, you really want to see this in the behavior in the movie. So this is slowed down, exhaling air bubbles, re-inhaling air bubbles as it searches for food. So when we look closely at these guys, we get to see that a number of these very unusual little mammals that are semi-aquatic have interesting specializations for olfaction, for example. But that's not the big story for this animal, because obviously it's got an interesting sense of smell, like the water shrew, with this ability to do this underwater. But the most interesting part of this animal is the star, right? So what the heck is going on with the star? Well, the star is an incredible structure. The mole is only a little bit bigger than a mouse. This star structure that you're seeing under the scanning electron microscope is about the size of your fingertip. So it's very small, but it's a remarkable structure. It's got 22 appendages. These are the nostrils that you saw the air bubbles coming out of. But it's got 100,000 large myelinated nerve fibers that are supplying the touch organs on this star. So I do want to say it's a touch structure. Although they're doing this underwater sniffing and they're smelling when they're outside of water also, the star itself, just like the surface of your own nose, is for touch, not for smell. The olfactory receptors are in the back of the nasal cavity, like you might think about a dog's nasal system. This structure is to give the star-nosed mole perhaps the greatest sense of touch of any animal in the animal kingdom. And so let me tell you a little bit about that. So as you go up in magnification, you can see that each one of these tiny little appendages is covered with thousands of little domes. And each one of those little domes is a touch organ called an Eimer's organ that has a series of nerve endings in it that are incredibly responsive to the slightest deflection when it presses its nose down against things. And I'll show you some of that behavior. But I did want to compare this also to a human hand, because humans have a great sense of touch. We have an expanded area in our brains that represents the hand. And part of why humans have been so successful is our ability to manipulate things, make stone tools, and use our iPhones, right? So that's part of why we rule the planet. So this is the star at the same scale as a human hand to give you an idea of how small it is. But the amazing thing to think about is there are 17,000 mechanosensory nerve fibers in our human hands. There's 5 times that number of nerve fibers in this tiny little structure the size of your fingertip. So contemplate that. Just try to imagine 5 times the sensitivity of your entire hand in the tip of one fingertip. and you can start to think about what it's like to be a star-nosed mole. And I do want to also emphasize that although it's a very bizarre structure when you look at it under the scanning electron microscope, when you get down into the details, this is a section of a human fingertip showing that the grooves that correspond to our fingerprints, essentially. And these are associated with touch receptors that are a lot like the star-nosed mole. This is at a different scale, but this is a star-nosed mole appendage showing you the anatomy of those little Eimer's organs, and this is a human fingertip. Not as different as you might think, and we are collaborating with investigators at the University of California and they are at Berkeley, and they are examining the molecules that mediate touch in mammals generally using star-nosed moles as a way to find where they upregulated and overexpressed. So that is a hunting strategy for these rare transduction molecules that may inform us about how mammal touch in general works, including in humans. So you can learn a lot of interesting things from these diverse animals, not just their specializations, but their generalities. Now, I wanted also to show you this animal now in real time, because I've mentioned the nose is for touch, and you saw it before as it was doing underwater sniffing. This mole has been trained to find this tiny little X on this piece of aluminum foil. And there's a food reward underneath that. And you'll see how quickly it goes touching things. As soon as it detects that little tactile cue, it breaks through and it gets a piece of an earthworm, which is one of its favorite things to eat. They love earthworms, and I'm going to sort of expand on that theme as well. It's shedding its winter coat, so you can see it looks a little shaggy there. So this guy was caught in the spring when things were warming up. And one of the interesting things about this animal that you might imagine, of course, is where is information from this elaborate touch organ going in the central nervous system. So interestingly, mammal brain organization has conserved features across lots of different species, including between star-nosed moles and humans, in the sense that the pathways to the neocortex are very similar for different structures. And you end up with maps of sensory surfaces. So the famous map in humans is the so-called Wilder-Penfield homunculus of the human body, laid out in the side part of neocortex for half your body, running from the midline down laterally, representing the feet, down to the face, and then the oral cavity. So we know a lot about that, and that was discovered through actually recording from human brains in awake subjects during surgeries, often for epilepsy. And so we can do similar recordings in a star-nosed mole and anesthetized preparation and map out where the brain area is found. And when you do that, you get a similar kind of thing. In all mammals, across from shrews to star-nosed moles to humans, you have a map of the body surface. Of course, it's got a mole shape with a big expanded area, the star, in star-nosed moles, whereas in humans, it's a big expanded area of the hand. But we can go a step further in this species and see some things that you can't see in many other species, and this is visible brain maps even at the neocortical level. So I want to show you that. So this is the neocortex from the left hemisphere of a mole in schematic form, showing a recording results. This is the anatomy of a neocortex that's been sectioned from the top down to look at the cellular structure across the neocortex where this information projects from different senses. And what you see here, this is where the star information projects. It's actually a visible star in the mole's brain, which is really remarkable and is very useful for a lot of experiments in understanding how touch is processed and how these different areas develop, how they're connected. I'm going to zoom in on this and show you for a different section here the closer-up view of the star representation in cortex. And here's the half star that goes with this cortex. So only half of the body is represented in each hemisphere of the brain. So half of the mole's star is represented in this part. Now, you can see there's 11 appendages and 11 subdivisions in this star. And it corresponds to where we get the responses from the star in cellular responses. And I said there's interesting things you can learn. There's something you can learn right off by looking at this anatomy, which is very interesting for this animal and one of the most amazing things about its organization for touch. And that is look at this subdivision here, this 11th subdivision. It's huge. It's much bigger than the appendage, the corresponding appendage. So it's expanded in the brain relative to its size on the star. And why is that true? Well, it turns out that that one part of the star is a touch fovea. And it's used the way we use our high-resolution central vision to explore things in detail. By that I mean, if I had you look at the corner of the screen here and try to read these numbers, you wouldn't be able to do it because your high-resolution fovea wouldn't be focused on the numbers. And star-nosed moles have the same kind of organization for touch. So they go through their world detecting things with these outer appendages and then making a sudden movement of the star very quickly that's very much like a movement of our eyes. These are visual saccades in primates, eye movements in primates. So it's amazing to think about how the organization of this unusual touch system parallels the organization of our visual systems in having high resolution and low resolution used for scanning, high resolution used for details. Let me show you the behavior of one of these moles, very much slowed down, touching something of interest, a piece of an earthworm, of course, with the side appendages, and then deciding, okay, I want to explore that in more detail, making a sudden star movement, and now it's touching it with the fovea to decide whether it's actually something it wants to eat or not. And that's a lot like our eye movements. And also, there are other species that do similar things. Amazingly, there is a bat, the mustache bat, that has an auditory fovea for sound processing of the echoes that it finds most important. And it uses that part of its central nervous system to process information that picks up the Doppler shifts of wing-beating moths. So there are auditory foveas in bats, we have visual foveas, and there's touch foveas in star-nosed moles. This does seem to be a general theme for how you organize a very high-resolution sensory surface. And that's one thing that gives star-nosed moles the ability to survive in their underground habitat. But there's another thing that they can do that I want to show you that is really remarkable. And this is related to their speed of foraging and detecting and making decisions. And this is something that everything else you've seen has been in slow motion except for that pulled back view of the aluminum foil test. Now I want to show you a mole filmed from below, and this is going to loop as it is detecting little tiny prey items and eating them. So this is incredibly fast. So the mole is able to, and it's actually going so fast in real time, you can't even see the foveation movements, but it makes one. For every piece of prey, it moves to the 11th appendage, decides whether it's something worth eating, and then eats it and moves on to the next thing. This turns out to be one of actually the fastest foraging mammals. And when we published that research, we got a call from the Guinness Book of World Records, and I didn't even really know what the conversation was about until this certificate showed up in the mail, which was just really amazing. You know, I sort of took the PhD off the wall and put the childhood dream. Even though I wasn't eating fast, it was the mole that was eating fast. So anyway, with these brain specializations, the star-nosed mole can really do remarkable things. And these are things that were unexpected at the outset of looking at this research. So I've shown you that the star-nosed mole is a very fast forager. And I went on to, and this is a lesson I'm learning over and over again. So I'll tell you what the idea was. I wanted to compare this amazing star-nosed mole with a run-of-the-mill, not-so-good forager, the common Eastern American mole. There's probably one within a half a mile of us right now. Maybe in a number of your yards you have these guys. Right, this guy, it does very well, lives throughout much of the southeastern United States. And the idea was it won't be able to find food fast because it doesn't have that special star, right? So I set out food items for it. And this animal amazed me by going directly from one food item to the other. It has no eyes. It can't see the food. And this looked like rapid olfactory localization. And the idea of how an animal could do this would be typically the way you might do the same thing in your house if maybe there was something burning somewhere. You might go from place to place, like where is that thing burning? You'd sniff. You'd say it's stronger here, weaker there, and you might be able to home in on the source of the smell that way. And on a smaller scale and more rapidly, that's something that this animal seems to be able to do. So let me show you this guy in action, sped up 2 times. This pieces of an earthworm with no scent trail, and it makes a beeline straight towards where the stimulus is, which is absolutely remarkable that they can be so accurate just by taking sniffs, moving their nose around. So good at this that even though they have no eye to speak of and a tiny little optic nerve, I went ahead and filmed it in infrared because I wanted to be 100% sure that they didn't magically use eyes covered in fur to do this. And of course they can't, but it's good to be thorough with these things. So under infrared they can't do it either. So really astounding abilities. And one of the possibilities there that has been thrown about for olfaction, and there is some evidence for, but it's kind of controversial, is could they be comparing the inputs to the 2 nostrils, sort of what you would call stereo smell. So we have stereo vision that gives us 3D images. We have stereo inputs to our ears that help us do auditory localization. It's been sort of an open, unknown question, just how many animals might, if any, use stereo smell. So this seemed like an interesting thing to address that, frankly, I think I started out thinking was very unlikely. I do want to mention that an inspiration for the basic study comes from owls, which have ears that are asymmetric, one ear facing up, the right ear facing up, the left ear facing down, and they compare, using stereo intensity cues from the 2 ears, those intensities to get the elevation of a sound source. And so one famous experiment by Knudsen and Kenishi was to put an earplug in to determine that, and to show that was to put an earplug in, and if you put an earplug in the left ear, it sounds louder in the right ear, and they make errors above them instead of accurately localizing the sound source. So what about moles? Well, you can anesthetize a mole, give it a temporary sort of cold, block up one nostril with a little piece of rubber, and ask, do they do the same thing? They do err on the side of the open nostril when they have one nostril blocked. So this is extremely strong evidence that they are using stereo cues as one of the additional facets of olfaction to get very quickly and accurately to an odor source. So this is examples of repeated trials where the animal, it used to go directly with both nostrils open straight to the food item, and it makes errors to the side of the open nostril, basically the intensity being stronger in that open nostril, sort of dragging it off to the side. But it has to be also using these serial sniffing cues because it still gets eventually to the food, eventually being still very quickly to the food item. So let me show you, because this is so extraordinary, I want to show it to you in a different way. Here is, instead of moving the food item around, showing you the same kind of behavior with a blocked right, a blocked left, or an open nostril, and just one trial where this is the normal behavior and this is the blocked right, this is the blocked left. You see that they are skewed off to either. So it's the same animal on different trials, and the averages show that you skew off to the side of the open nostril. So absolutely remarkable that this animal can do this, and sort of the conclusion for this species is that it is seeming to use both serial sampling, going from place to place and looking at stronger and weaker smell, and this bilateral sniffing, especially the bilateral part when it gets close, the stereo sniffing that helps it get home in at the last minute on things. And I also want to mention that if you can do this for something that's just out there as an odor source within a gradient, what might you be able to do with a scent trail, right? That seems like a much easier task. And this is an example sped up of no scent trail on the circle, and then an earthworm scent trail laid on the circle. And you can guess that this guy's pretty much obsessed with that scent trail. So probably has a better sense of smell than most other mammals, because this is not something that has been identified in other mammals. And one of the things it means is if you're an earthworm, you are in big trouble if this guy gets on your trail, right? They love earthworms. In fact, they can eat 7,000 of these things a year. 5 times this number of earthworms. So there's a huge selective pressure on earthworms to not get eaten by moles. And maybe they can come up with some way to deal with this. And this is one of the themes in the research, is always these predator-prey interactions, right? Well, this is actually a friend of mine now, Gary Rebel, who has a very interesting job. He does something for a living called worm grunting, and that is this mysterious and legendary sort of bait collecting technique in the southeast, particularly in the Apalachicola National Forest in Florida, where for generations they have been collecting bait by pounding a stick into the soil and then rubbing that stick with a piece of metal, causing really strong vibrations in the soil, which causes thousands of earthworms to come to the surface. It's an amazing thing to see. They actually have an annual worm grunting festival, and you can come out and try your hand at this. The kids can come out, and the winner gets a prize. They have a worm grunting dance, and they crown a worm grunting queen. It's a really kind of a fun thing to see. And they're trying to keep their tradition alive and educate people about the history of the area. But let me show you them in action. So this is Gary Rebel. He allowed me to come along as they do their normal work. It's the vibrations. They don't have ears. Yeah, but it's a good question. It's a good question, because many species of insects have ears, for example.
I had to make a fisherman leave home,
Yeah.
you know.
Wow.
That's a pile of them.
Somebody asked, why did they come to the surface? That's the key question. They've never known. This has been a mystery, right? There's been lots of theories. And one of the main theories was, you might guess, rain, right? So worms do come to the surface after a long rain, but they don't bolt to the surface at the first signs of rain. And in fact, if you put hundreds of these specific earthworms from the area in a bin and then wait for a storm, and this is the actual radar trace of the storm that was for this test, after an hour, an average of 2 earthworms comes out. But if you put a mole in with them, you'll see something different. The worms are avoiding the vibrations caused by digging moles. Turns out there's a huge number of moles in the area where the worm grunters do their work. And these worms have evolved an escape response to the oncoming mole. And if I could ever describe a worm as running, this would be the time. Now, they can do this because the mole will not come to the surface to chase its dinner on the surface. And the reason for that, you can imagine, it's chasing its dinner. The worm's about to be eaten. It should take the chance to come to the surface. But the mole's only trying to get dinner. And it shouldn't take the chance, because it has its own predators that will go after it if every time that it was trying to eat, it came to the surface. As a blind burrowing animal, that would not be a wise strategy. But if you're about to die, like the worm, then it's the best choice that you have. And unfortunately, you might end up coming up to Gary Rebel and ending up on a fishhook instead. Now, the first person to propose that this might be going on was Charles Darwin. So he wrote, of course, Origin of Species, but he also wrote, closer to his death in 1881, after the Origin of Species, this wonderful little book on earthworms. And in it, he pointed out that it has often been said that if the ground is beaten or otherwise made to tremble, worms believe that they are pursued by a mole and leave their burrows. So there's this interesting historical footnote. He never really was able to demonstrate this because he had a couple other things on his mind. But it turns out he was right about that, and he wasn't the only person who's suggested this. So it turns out, and these are great, interesting predator-prey interactions involving multiple species, that wood turtles beat on the ground as a way to get earthworms to surface, and then they eat them. And the person, John Kaufman, who studied this in Florida, was aware of worm grunting and actually said that this was a form of worm grunting. And another very interesting account of this sort of behavior is foot paddling in seagulls. So Nico Tinbergen, a famous ethologist, wrote this wonderful book on herring gulls. And in that book, he pointed out that paddling has two different functions. One is bringing up of earthworms, which seem to have an innate reaction to the quivering of the soil, which is of value, enabling them to escape their arch enemy, the mole. So this idea has been out there for a while, but never really formally tested. Now, I told some folks about this at a talk, and someone in the audience, Jakob Christensen Dalsgaard, actually was aware of foot paddling and saw it going on outside of his office in Denmark. And he then went home and filmed the seagull foot paddling and eating earthworms, which I thought was just really kind of amazing. And what was really amazing was after watching the seagull for a while and sort of sending me this clip, he pans out, and there's the mole tunnels in the background. So the seagull is essentially worm grunting right around the moles where the earthworms are escaping from the vibrations of moles, and it's pretending to be a mole to take advantage of that escape response. And this also kind of explains something that I couldn't resist showing you, which is this rather grisly finding by a naturalist on a beach in Europe, which is basically a seagull with a mole emerging from its chest. And it didn't take much forensics and sort of CSI to figure out what had happened there. So this is why, as a mole, you don't pursue the earthworms to the surface, right? So this seagull ate this mole, clearly, but the mole was still alive. And the mole didn't like where it was and burrowed its way out of the seagull. And they both died, so it didn't fully escape. And so I think the lesson there is that you should chew your food, really. So this idea that you can tap into the behaviors of prey that are normally adaptive, right? So normally it's a good idea for an earthworm to come to the surface when it senses vibrations, because there's a lot more moles than there are worm grunters. There's more moles than there are seagulls in general. So you can take advantage of this, and this is something called the rare enemy effect. As long as you're not too common, then you can sort of deceive your prey in interesting ways. And I want to continue with that theme and tell you about this guy, which is a tentacled snake. And he looks really, really intimidating and scary under the scanning electron microscope, but really it's an innocuous small water snake that's totally harmless. So in its natural habitat underwater, it would assume this unusual hunting posture and wait for fish, holding very still, hoping that they come close enough to strike at them. And that's all it eats. It's totally aquatic, never comes out of the water, gives birth, live birth in water to its young in water, and only eats fish. So in areas of Southeast Asia where this animal lives, it's called the fishing snake, and it's an expert at catching fish. And this is interesting because I mentioned that fish are remarkable escape artists and are very fast, and they are. In fact, they are a heavily studied model system in the neurosciences, basically looking at the very large neurons that efficiently generate these rapid turns and swimming away from predators in fish. This is something called a sea start, and interestingly, the time it takes for the snake to strike out to the position of a fish and the time it takes a fish to start to escape are about the same. So these two behaviors are intertwined. If you're a tentacled snake striking at a fish, the fish is inevitably going to trigger this super fast escape response to try to get away. So that sets the stage for some interesting coevolution. And the C-start, the escape response, can only occur in one of two directions. You're either going to turn left or you're going to turn right. And it is directional, meaning that when the fish detects a water disturbance on, for example, the right side, it will turn to the left and vice versa. And it does this through an auditory system. So got the question about earthworm ears. Well, fish actually surprisingly do have ears. And they're very sensitive to underwater sounds, which one important sound would be the sudden sort of shock wave of a striking predator, which inevitably creates a lot of water disturbances. So let me show you a C-start. This is filmed at 2,000 frames per second. And it's slowed down. There's the water droplet. And then the fish bends into a C shape. And I won't show you the data for this, but it's definitely not a visual response. Vision is actually not fast enough to mediate that response because of delays in the retina. So this is a response to the propagated pressure wave in the water, and the time from that water droplet touching the surface of the water to this fish moving in this trial was 5.5 milliseconds. That's very close to about a 200th of a second. So just to give you an idea of how incredibly fast fish are, they're remarkably fast. And we know a lot about how they do this. So there is a very interesting neural circuit that the fish use. And this is one reason that they've become a model system for studying neural circuitry. And there's been a lot of interesting discoveries in this circuit about the way synapses work, about electrical versus chemical synapses, about large fibers, about inhibition of action potentials by some neurons. So let me just tell you briefly about this circuit. So you've got the ear, you've got hair cells that detect the movements that pass this information onto a neuron in the 8th cranial nerve, which is an auditory neuron. This projects to a giant cell called a Mauthner cell. So that's one thing that's really interesting. It's a huge neuron in the fish brainstem. They're paired. There's only two of them. And this cell projects to the opposite side of the fish body, projects information down from there to cells in green here that activate the muscles. And so what happens is this all happens very quickly, partly because there's electrical communication in different places of this circuit. So if there's a water disturbance on this side of the fish, very rapidly these muscles contract as a result of the activation of this circuit, and the fish moves this way. But you might imagine that if you're going to do that, you want to make sure that you inhibit the muscles on this side, right? If you were to simultaneously contract both sides, maybe this Mauthner neuron might fire because of sound impinging on this side. If you don't inhibit this side, you can't make an efficient response. You also want to inhibit any ongoing activity, like maybe you're swimming. So this red cell sends inhibition, which turns off the motor neurons on this side, turns off the muscles on this side. So that's sort of in a nutshell, a very fast overview of how this works. And I want to tell you what the snake has figured out. So the snake seems to have got a diagram of the fish neural circuit and has studied up on it. So first let me show you in real time what a tentacled snake strike looks like, because they're just so fast. And the main purpose of this is to just show you that you can't see what happens. It's too fast to see without slow motion. But let me show you what happens if you slow things down. And what I'm going to show you is a remarkable trick. And the trick is that about a millisecond before you strike out with your head, you move this part of your body. And what that does is trigger the escape circuitry in the wrong direction. And because of that inhibitory cell, once you start turning in the wrong direction, you cannot go back. You're committed to turning away from that attack, which isn't actually the attack in this case. So I want to show you the snake in action. This is slowed down immensely, 1,000 frames per second. And this explains descriptions before there was good high-speed video of how quickly this snake feeds on fish, that the fish just sometimes totally disappear during the strike. And how could that be possible? Well, if you swim into the snake's mouth, that helps the snake. Sometimes they turn in the correct direction. It's not a perfect trick.
I was going to say, how do you know the same thing's not happening in the snake's body that's happening in the fish body? You know, with that.
I'm not sure.
Do they not have ears? Or you know how?
Oh, you mean to try to escape?
And it's on the other. It activates the trunk muscle.
Yeah.
So does you know that the snake is doing the same thing, activating
Its own movements? Well, it is actually can go either can bend in either direction depending on where the fish is. So it doesn't strike in either direction, but it'll set up its attack in either direction. And it's a very visual, so it will use vision to guide this behavior. So it knows which side that fish is coming at. It's not using the sound of the fish. It's using vision. But it has another trick. Actually, let me just show you the data here in a little more detail for that movement. So here you're going to see this first part of the strike just to show you how subtle that movement is. This is looping back and forth. It doesn't actually vibrate. But that little tiny movement actually generates the pressure wave. And that's enough to skew the fish to move in the wrong direction. Right? So it's really kind of hacked into that neural circuitry. So they exploit the C-start, and that is a rare predator effect. But that works really well, right, if the fish is in this direction, right? But what if the fish is approaching you from this side? And if you're getting approached directly, and the fish can either turn left or right, you can't scare it forward into your mouth. And when the snake encounters this orientation, it does something that I think is even more impressive than the sort of tricky directional trick, and that is a predictive strike. So it does the same thing with its body. The fish then escapes from that movement, and the strike comes to where the fish is going to be in the future. So this is so fast that the snake can't change its direction of strike once it has started. So what you're going to see is a fish, and then a mask appears over the fish so you can see its original orientation and see that the snake is aimed for where the fish head is going to be. So it's basically planning ahead, in a sense, to where the prey will end up in the future. It pulls its eyes in when it strikes, and it is you saw those real-time videos. It's a ballistic strike with no possibility of adjusting on the fly for where the fish is actually moving. So it has to decide ahead of time. And remarkably, these snakes are born with this ability. So it's not learned. They don't learn this from a lifetime of striking at escaping fish. They'll do this from the time that they're first born. A question back there.
Is there any evidence that the fish is developing or evolving a defense against this snake?
I mean, that is a great question. And the question was, is there any evidence that the fish are evolving a counter strategy? And we haven't seen any evidence of that, but we wouldn't probably know because we don't know much about what's going on in the natural habitat in sort of Thailand where these guys live. But you would predict probably not, mainly because, well, I might say, have any of you ever heard of a tentacle snake before today, right? And I don't think most fish have either. And basically, there's a huge number of snakes in the area. And one of the sad things to see is sort of the fishermen come back with a haul of snakes to sell for food, and it's just a pile of snakes. Only a small number of those are tentacled snakes. And so the premise would be that they're using this sort of rare predator strategy. And the idea is that the best thing for the fish to do is flee as quickly as possible away from the water disturbance, because 99 times out of 100 or more, it's not going to be a tentacled snake. And if you started sort of tweaking the system to go in the other direction, most of the time you'd be wrong. That's the idea behind it. Of course, it'd be nice to get more information, and maybe you could have, even in the lab, have a bunch of fish that got selected to not do this. I will tell you something really interesting. So I've for the juveniles, because I didn't want them to learn during the experiments, would hand feed them with tweezers dead fish, and they would strike to the side of the fish, trying to get the fish to move. It was really just remarkable to have the experience. Of course, there's enough variability that you can get them to eventually get a hold of the fish, but it's fun to watch that ingrained behavior as they do that. Okay, so the last thing I was going to talk about was the revenge of the fish. And this is a special fish. It is a fish, and it's an electric eel. And electric eels are our latest study subjects in the lab, and they're absolutely remarkable. So to tell you about electric eels, they live in South America. They get quite big. They're somewhat famous. And I want to start the story about how could you possibly end up being an animal that generates electricity, right? So that's a weird thing to be able to do, and this is something that really troubled Charles Darwin about intermediate forms of electric sense, or I'm sorry, about generating strongly electric fields. Like, what good is it to have a weak electric field if you can't use it as a weapon? Well, we've learned a lot since that time about why you might generate electric fields. And it turns out, I would say that the start of the story is from the sense of electroreception, the ability to detect electric fields in your environment, something that is characteristic of sharks, amphibians, at least one mammal. It's a very interesting sensory ability, and it's a passive ability to detect an electric field that is generated by some animal or object that is out in the environment. And one thing to know is it's incredibly sensitive. So the ability to detect these electric fields, which great white sharks, for example, have, is detecting primarily the electric field given off by, say, prey items that is very weak, but there. And you might be aware of this if you think about what a heart monitor does, right? You're aware that we give off electricity based on muscle movements, muscle contractions. Leakage of ions in the water from prey also does that. Nerve and muscle contractions, movement of gills, these give off very weak electric fields, and they're specialized organs that are electroreceptive, connected to the central nervous system that sharks use for this. And this was considered sort of a classic set of studies for sharks by Odd Calmeen that basically identified this sense through detailed behavior studies combined with electrophysiology and anatomy. And actually, one interesting thing, I'll just as an aside, that I thought was sort of fun to follow was that since the discovery of this sense, there are now companies that will sell you a shark shield that is basically based on the ability to deter sharks by overstimulating their sensitive electroreceptors. So that is something that some divers now wear to protect themselves from sharks. And again, the sense of electroreception sort of builds on the premise that muscles generate electric impulses. And here's our most common sort of day-to-day knowledge of that. You know, you think of somebody wired with electrodes on the surface of your chest that will be able to detect the electric fields generated by muscle contraction. Now, you can sort of elaborate from that to imagine something else that has happened in some species of fish. So some species of fish have gone a step further, and these are African knifefish, South American knifefish, they're gymnotid fish and mormyrid fish, and they generate electric fields with a little organ in their tail. And this is used as a radar system to basically probe their environments. And sort of the idea is, what most likely happened there is they had passive electroreception. They were detecting muscle movements and other fields, and they could detect their own muscle movements. And that allowed them to modify their own muscles into batteries that now they use to generate just electricity for navigation. And most of these fish are little delicate knife fish that are nocturnal and feed on small invertebrates, come out at night, and you can often see these in pet stores. But there are some exceptions, and this is one of those exceptions. This is sort of the going down the weapons of mass destruction pathway for your electro-generating muscles, or your modified muscles, which are electrocytes. So that's one thing that I think is a very interesting system. So this is the electric eel. It gets very, very large, and it is actually a modified or evolutionarily derived knife fish. So it's not related to the other eels you might think of. It is a relative of those little electric fish, in this case the ones from South America, the gymnotid fish. And it gets huge, right? Here's somebody holding one of these big ones, and it has basically converted much of its body into an electric organ that generates electricity. And this is where all the internal organs are. The rest of this, except for some swimming muscles, is modified muscles that now generate electricity, these electrocytes. So I'm going to show you this in action. So this will be an electric eel, and you see the fish, that's the prey. So that was it giving off a very high voltage attack pulse to essentially deactivate the prey. And how does it do this? So this is a very interesting ability. So this is that sound you heard, that sound, that was a series of pulses given off by the electric organs of the eel. And if you're a prey fish that it's nearby, essentially you get frozen in place. So what do I mean by that? So this is shown schematically here over time. The red plates are when the electric discharge starts. But let me show you that in a video, because it's much more easy to tell what's going on. So this is slow motion, and when you see the red flickering, that's the electric discharge. And the fish is just frozen. And the question is, of course, how could they possibly do that? The other thing I should mention is, again, this is happening very quickly. The fish is frozen in place in about 3 to 4 milliseconds. It's exactly what it is. That's exactly what it is. So how does this work? Well, it turns out you can do the experiments where actually you anesthetize a fish and then you sacrifice it and actually remove its brain. So there's no brain left in these fish from these experiments. It's just a muscle preparation. And when a nearby eel gives off its discharge, all the muscles in the fish contract. They essentially freeze up the fish through tetanus, and that is blocked by curare, which tells us that it's basically activating the motor neurons of the prey. And it is a taser. It's essentially doing what a taser does, except a taser has a 19 hertz frequency that stimulates muscles to deactivate people that may be an assailant for police, right? And in a sense, you have modified muscles that the fish, the eel, is using, but look at the discharge rate compared to a taser. So it's not showing any restraint. Of course, a taser's engineered so that you don't overcontract your muscles and get injured. The eel is not so worried about that. And if you read the historical accounts, it turns out that, in fact, people are immobilized by eels. If they step in the wrong place, and this is an account historically about natives in an area where the eels were, in the 1800s, they were collecting eels. If they stepped out of the boat and an eel gave a discharge, they were just stuck there. They couldn't get into the boat. They couldn't get out of the boat. And that's what happens when you step near an eel. Now, in closing, I also wanted to mention that electric eels, combined with the venom of the banded crate, were incredibly useful in identifying the acetylcholine receptor, which is the basis of muscle contraction in mammals. And so these guys, and why was that true? Because if you're going to generate huge currents with your muscles. You need to amplify the number of channels that are allowing ions to pass back and forth to generate high voltages. And so that made for a very useful system for investigators to examine where and what are the channels before they knew what they were. So historically, during the 70s and around that time, electric eels and other electric fish, such as the torpedo ray, were used in conjunction with venom that binds to the channels to figure out what they were. So a lot of these animals end up actually showing us really fundamental principles about how nervous systems work, how neurons work. And I want to close just by thanking many people who have worked on different projects from my lab. So we've had postdocs, graduate students, all working on different species, star-nosed moles for Sam Krish and Fiona Rempel. Christine Krish actually worked on naked mole rats, which I didn't talk about. Eva's worked on the brainstem of water shrews and star-nosed moles. Duncan worked on alligators and crocodiles, which I didn't have time to talk to you about, but also have a fascinating sensory system. Paul Morosco did a lot of electrophysiology in star-nosed moles, showing how those Eimer's organs work. And, of course, the worm grunters showed us how worm grunting works, and were very generous in taking us around that area. And my wife, Elizabeth Catania, also works on a number of these studies in the laboratory, including star-nosed moles, water shrews, and other species. I also want to thank a number of our funding sources and the Department of Animal Care and Oversight at Vanderbilt for helping us troubleshoot how to house so many weird and unusual animals. Thank you very much, and I'll take questions. Yeah, question over here. I mean, that's a very interesting question, whether earthquakes might affect these escape responses. And I don't know of any, but I wouldn't be surprised, because there are other people studying vibrational responses. I wouldn't be surprised if somebody's followed that up in some countries. But I don't personally know about that.
Which year you were part of this study? Which year? How long ago? The study?
When did we start these studies? Wow, I first started looking at some of these species back in the late 80s when I used to work at the National Zoo in Washington, and have just continued to look at different species since then through graduate school, and then taking a position here in 1995.
So who was the first person to find out?
Find out what? Find out what?
Is that related to the animals? Is that human? How you study, how you train?
Well, for example, something like the electric eels have fascinated people for thousands of years. Electric catfish, they're carved into Egyptian stone about what fish were available, and we clearly have accounts of them being used for attempted cures of migraines and that sort of thing. So people have had an interest in these unusual animals for thousands of years, and certainly for centuries, people have wondered about a lot of these different aspects. Yeah. 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 Yeah, so basically that is through recording experiments very often. So to go in and record in an anesthetized animal how the neurons are responding, combined with anatomy. So you can put tracers in to look at how the circuitry is organized. So that's one of the ways that people have explored, for example, the fish circuitry in such great detail. And then there's genetic manipulations in zebrafish, for example, that can tell you a lot about what goes on with that kind of circuitry. So there's lots of different approaches that can be used. And in a way, it's easier than to understand human brains because, of course, we're willing to do experiments where we record and look at anatomy, where you can't do those kinds of things in humans unless it's a surgical situation. So a whole range of different ways of approaching. Is that some help? Yeah.
You mentioned the one-to-one mapping.
Yeah.
And illustrated it so dramatically.
Yeah.
Errors, I'm sure. Is this key to vision or other senses? if you screw up those connections,
Yeah.
do you adapt to that and see just fine? I heard people putting on glasses and making stuff like that.
Yeah. That's a great question and a really interesting area of study. So the idea is basically what goes on when you alter these maps, when there's damage to these maps. A related question might be what happens if you lose input to a map. Maybe you lose an arm, for example, things like that that happen, unfortunately, during combat. So there is a lot of plasticity to these things. And there's sort of been a revolution, I'd say, sort of starting in the 90s, in us understanding that even adult brains are really malleable, much more malleable than was thought. So it used to be thought, okay, you have these critical periods, and then after that, we're all kind of stuck. But that's not the way it is. So when you look at the experiments, one of the great places where this was looked at, I mentioned the barn owls, for example. They've been a key study species for how you integrate and align maps of vision and sound to generate appropriate head movements. And it turns out that if you put an earplug, like I showed, for an owl, they'll adapt to that earplug and correct the auditory movements so that they can adapt to that earplug. If you put vision shifting goggles on the owls, they will be able to adjust to those vision shifting goggles by changing their auditory system. And they do this partly through growing new connections in their brain to rewire areas. And those new connections are very interesting because in some cases they will allow the animal to adapt to things in the future that relate to the, so once the goggle experience has happened, they can then readapt to that goggle experience. Here at Vanderbilt, people have studied primate brain reorganization related to, for example, phantom limbs. So one of the things that has been found, and this is in the Koss laboratory here in psychology, is that there's actually sprouting of new connections in the primate brainstem from the face region to the hand region when you have the loss of inputs from an arm. And that explains how phantom limb sensations can be generated from touching the face, which is a common reported experience for humans. So a lot of these maps are very malleable, and changes to the maps occur. Filling in of maps occurs from adjacent areas. New connections grow from adjacent areas. So things are quite plastic. Yeah?
Those were done in adult animals?
Those were done in adult animals, yes. So there is lots that can happen in adult brains. I will say an interesting study, in terms of barn owls, that, I like to talk about some of the specialized species, but they are a mainstream model system for this plasticity in the central nervous system. It was once thought that if you put on shifting goggles on an adult, they could never adapt to the shifting goggles, and only the juveniles would adapt their auditory system to the shifting goggles. But then the experiment that was done was to, instead of shift with a large shift in the adult little by little. And by doing that, training little by little, they got the same degree of adaptation that a juvenile with a lot of plasticity could have. So the theme there was, don't try to make a giant leap at one time, but you can do a lot and decide that because it didn't work, it's impossible to learn this thing. Going incrementally in training may get you to the same end result through experience. So there's a lot of new information about that.
Does this work have implications for persons who have strokes?
Absolutely, it does. So some of the early studies that were examining brain reorganization related to strokes. So, I wouldn't say my work has that I just presented, but these areas of examining brain plasticity definitely relate to that. So how does the brain recover? What happens when you lose an area? So what's been shown in a number of primate studies, and some of this work is by a guy named Mike Merznick and others who have simulated strokes in primates and then looked at what happens to the brain maps, and it turns out that some brain maps will change their function to compensate for the lost part of the map that has been damaged. Other areas will fill in with new inputs that will also compensate. So the way those experiments were done was to simulate a stroke by damaging an area of the brain after an animal had been trained to do a complex task. And as you might expect, the area of the map related to that task, they were no longer able to accomplish that task. But then the animals retrained through this kind of repeated learning and activation, and after they regained the ability to perform the task, it was found that these maps had reorganized in the adults. So there is a lot of interesting plasticity in the adult brain that's going on, and a lot of new research in that area.
My dog has an uncanny ability to hunt moles.
Oh, how interesting.
If we were on a walk, he will all of a sudden stop dead, stand at a certain spot, and triangulate.
Uh-huh. Uh-huh.
And if I let him go, I often don't let him go unless he's in my yard. He will dig, like fast.
Yeah.
And bop the mole with his nose.
Uh-huh.
And stun it and pull it out.
He's a good hunter. Yeah. I would love to know. I have to say that I think dog olfaction is a goldmine of untapped, interesting behavior. So we have dogs as well, and they have incredible olfactory abilities. They can follow people, along a path quite some time directionally after someone has walked somewhere. They might have this stereo smell ability. They just have an incredible olfactory system, and yet I don't see many papers that have examined that, and it might be pretty easy to do with some dogs are so trainable. It wouldn't probably take too much to try to learn some really interesting things. I would think so. Yeah, actually, I probably have spent an above-average amount of time around mole tunnels, and there's absolutely no doubt that you can hear them digging. So I actually have a video, which I didn't have time to show, that demonstrates why a worm would be able to sense the vibrations. And this was in Tennessee. I just had the microphone on a video camera on a tripod, and you could pick up the sound of breaking roots. They're really powerful. Sometimes when we were trying to do these olfactory experiments, we'd go to the golf courses where that's where they hate moles and they want you to come collect the moles. So, you'd stand around trying to collect the moles, and you can hear them as they're extending their tunnels. Sometimes you'd be looking, I think it's over there, and you'd hear it, and it's right behind you. So I bet the dog is cueing into that. Yeah. It sounds like a good hunter. You're welcome.